so much. Honorable Chairpersons and organizers of the International Conference and Expo on Medical Microbiology by Magnus Conferences, I am Dr. Manisha Mandal, Assistant Professor from the Department of Physiology, MGM Medical College, India. The topic that I am going to present before you is titled Studies on Alteration of Gut Microbial Composition with Probiotics Administration in Health and Disease using Metagenomic Analysis. Now the background of the study. Functional foods including probiotics have attracted increased attention in many countries. Probiotics are live microorganisms that when ingested in sufficient quantities confer health benefits. The gut microbiome, an immensely diverse and dynamic niche of the human body, is a critical determinant of human health and disease and a key regulator of host physiology. But it is not clearly known to what extent the ingested probiotics affect the composition and functionality of the gut microbiota. Since this study deals with probiotics, so it is important to discuss the mechanism of action of probiotics conferring health beneficial activity to the host. Firstly, probiotics modulate immune system by enhancing antibody response. It also reduces inflammation and it also causes stimulation of phagocytosis. Besides, probiotics interact with gut microbiota through production of antimicrobials by cross-feeding and substrate transformation and supporting microbiota stability. In addition, probiotics produce organic acids like lactic acid, propionic acid, and acetic acid, thus reducing colonic pH. It also increases butyric acid production through cross-feeding. It also confers colonization resistance by competing for nutrients and location. In addition, probiotics improve barrier function by stimulating mucin production and supporting epithelial cell health. In addition, probiotics interact with the host mediated by cell surface structures such as pili LPXTG binding proteins, mucin binding protein, toll-like receptor ligands, lipotecoic acid, and exopolysaccharides. Probiotics also help in the synthesis of small molecules having systemic effects like neurochemicals such as cortisol, serotonin, and gamma aminobutyric acid. It also synthesizes tryptophan and histamine derivatives, satiety hormones, and conjugated linoleic acid. In addition, it produces enzymes such as bile salt, hydrolases, and lactases. So these are the diverse mechanisms which drive probiotic beneficial effect to the health of the host. So with this background, the present study was carried out to understand probiotic food benefits by metagenomic analysis of 16S ribosomal RNA gene marker from human gut microbiome. The method adopted to achieve these objectives were ME6 single-end FASTQ sequences of 16S ribosomal RNA bacterial genes were retrieved from SRA toolkit NCBI pertaining to 25 gut samples from 5 datasets in health and disease, including those with probiotic administration comprising 5 healthy controls, 3 sets of type 2 diabetes mellitus cases, 3 without type 2 diabetes mellitus, 4 cases of Prader-Willi syndrome, 4 cases of obesity, and those without probiotic administration comprised 3 cases of healthy vegetarians and 3 cases of healthy omnivorous. The FASTQ sequences were imported to KIND2 in Miniconda 3 and subjected to demultiplexing quality control with DARA2 algorithm, phylogenetic analysis, and taxonomic classification using Green Genes database. The operational taxonomic unit abundance were attached with their corresponding taxonomy to generate the biome file, and the downstream statistical analysis as well as the data visualization was carried out using microbiome analyst. Community profiling was done with alpha diversity, beta diversity, and core microbiome analysis. Clustering analysis was achieved with heat map to compare abundances between different taxonomic levels, dendrogram, correlation analysis, pattern search. Differential abundance analysis was done with univariate statistics, and marker gene survey was carried out using metagenome sequence package. The PICRUST was used for the prediction of functional potential and pathway analysis of the microbiota from 16S output. Now the results. So this slide represents the alpha diversity of OTU in gut microbiome after probiotic administration measured using cow1 index and ANOVA, from which the values were F value were 20 and P value was highly significant, which showed association between microbiome structure with probiotic administration in disease and health conditions. Here, the graph shows that the healthy omnivorous individuals demonstrated higher alpha diversity compared to healthy vegetarians. Obese individuals, they showed higher diversity compared to type 2 diabetes mellitus, followed by Prader-Willi syndrome. And type 2 diabetes mellitus, those individuals were more diverse structurally compared to their non-diabetic counterparts. And this is the rarefaction curve, which shows species richness. It shows that in health, obesity, and in type 2 diabetes, species richness was more evenly distributed, whereas healthy omnivorous individuals were less species rich compared to the healthy vegetarian individuals. And this plot shows beta diversity, which has F value of 1.89 and a P value also significant, which shows similar variance between two principal components and three main confidence ellipses comprising healthy individuals, obese, and Prader-Willi syndrome. And this is the relative abundance of gut microbiome in health and disease with probiotic administration at phylum level, which shows a percentage of 46 for Firmicutes, 44 for Bacteroidetes, and 5 and approximately 4% of Actinobacteria and Proteobacteria respectively. And this is the relative abundance at class level, which shows the presence of approximately 42% Clostridia, 35% Bacteroidea, 10% belong to the unassigned group, 4% of Actinobacteria, 2% Gamma Proteobacteria, and less than 2% of Escherichia, Bacilli, Beta Proteobacteria, and Corybacteria. And this is the relative abundance at order level, which shows the presence of Clostridials, Bacteroidals, Bifidobacterials, Enterobacterials, and less than 2% of the rest as shown here, and their corresponding percentage here in the right column. And this is the taxonomic classification at the family level, which also indicates the presence of Lachinospiraceae, Ruminococcaceae, Bifidobacteriaceae, Ricenellaceae, Enterobacteriaceae, and less than 2% of the families indicated here. Again, at genus level, there were Faecalibacterium, Clostridium, Bifidobacterium, Alistipes, Ruminococcus, Gemiger, Doria, Butyribio, and Olsinella. And at species level, Bifidium, Onderdonkey, Lactaris, Saccharogumia, Formicillis, Longicatena, Ruminatium, and Chrysotos were found. And this is the phylogenetic tree of the species at family level, showing evolutionary relationship between the microorganism, whereby Escherichia, this was more distantly related than the rest of the other members of the family. And Corybacteriaceae, it diverged from the ancestral lineage of all the remaining families. And this plot shows univariate analysis at OTU level, showing abundance in terms of log transform count across different cases, exhibiting enrichment of this OTU number showing here representing clostridials. Again, another number with clostridials, and here choreobacterials between the different cases. And this represents the functional diversity in terms of log KO counts between the samples. The functional diversity and functional richness, it shows that type 2 diabetes mellitus was functionally more diverse compared to those not having diabetes. While type 2 diabetes and Prader-Willi syndrome, they were functionally more diverse compared to healthy counterparts with probiotic administration. And healthy vegetarians were functionally more rich compared to the healthy omnivorous irrespective of probiotic administration here. And this slide shows the relative abundance of KEG metabolism from Picrust analysis of 16S ribosomal RNA output. So it shows here expression of metabolism related to amino acid, to carbohydrate, energy, lipid, cofactors, vitamins, amino acids, terpenoids, polyketoids, nucleotide, resides, biosynthesis of secondary metabolites, and xenobiotic biodegradation as well as their metabolism. And this slide demonstrates the comparative Venn diagram corresponding to probiotic treated specific and shared function at KEG ortholog level in health and disease. Now, the conclusion. Post-probiotic treatment among type 2 diabetes mellitus, Prader-Willi syndrome, and obese individuals expressed lactospiraceae as the dominant family known to be involved in the production of short-chain fatty acids compared to healthy individuals dominated with Ruminococcaceae known to be more abundant in a stable gut microbiota during probiotic treatment. Bacteroidetes, which is highly relevant in dysbiosis and disease, was significantly reduced with probiotic administration in type 2 diabetes mellitus, in Prader-Willi syndrome, obese, and was eliminated from otherwise healthy individuals. Enterobacteriaceae, responsible for causing nosocomial and community-acquired infection, were completely removed from healthy individuals with probiotic intervention, while suppression of Enterobacteriaceae was comparatively more effective in type 2 diabetes mellitus than Prader-Willi syndrome and obesity. There was an abundance of beneficial bacteria, Bifidobacterium brevi, with probiotic intake more in Prader-Willi syndrome compared to healthy type 2 diabetes mellitus and obese people. So that was all for my presentation. Thank you for your patience in hearing. Thank you.